Pleiotropy is where you have one gene that causes many changes in the phenotype. So you could ask, like, how could a single mutation affect multiple parts of the body? And the good example of this, kind of the best example of this, is Marfan's syndrome. So people that have Marfan syndrome have just a single mutation in a gene that codes for a really important protein in the connective tissue. So the gene codes for a protein called fibrillin, and this is important in the development of connective tissue. Right? And it is autosomal recessive, which means that you really have to have 2 copies of the mutant allele in order to have Marfan syndrome. Okay. The thing about Marfan syndrome that's interesting is oftentimes people do not realize they have it. And so we actually had, in the past, a president of the United States that had Marfan syndrome. Does anybody know what president that was? So if we look at the phenotypes of Marfan syndrome, the phenotypes are long limbs, fingers and toes. So they tend to be pretty tall, right? They have eye problems. So they don't see very well. And they also have heart problems. Okay. So the president that had Marfan syndrome, or is thought to have Marfan syndrome, was Lincoln. Okay. So Lincoln is thought to have had this. And so it's really important that people get diagnosed with it, because if they're not diagnosed, then they could possibly maybe have a major heart attack when they're at a relatively young age because of the heart problems that is due to this. And oftentimes it just goes kind of undetected until they're adults and they start to have problems with that. And kind of interestingly, it gets worse as a person ages. So it might not be that much of a problem as young people, but as they age, they tend to get worse symptoms of it. Okay, so that's a good example of Marfan syndrome, and this is a diagram of maybe a person that might have Marfan syndrome. Okay. So another thing that's really important to consider is that we can have some traits that are not determined by a single gene or even 2 genes, but a lot of genes. And so this is what is referred to as polygenic inheritance. So this is where you have many genes, not just 2, but many genes determining the phenotype of an individual. So one interesting example is skin color in humans. And in skin color, there's at least 6 genes. Now our coloration, our pigment, is due to the presence of melanin. So color due to the presence of melanin. So melanin is a pigment that actually also determines your eye color, but it's in your hair, right? And it's also in your skin. And the more melanin you have, the darker your skin will be. And actually skin color is also a good example of how the environment affects the phenotype, because if we are out in the sun a lot, our skin will turn darker in order to protect ourselves from ultraviolet radiation, right? So it also has an environmental influence, okay? So I'm going to use just a 3-gene model to talk about the inheritance of skin color. And so the genes are A, B, and C, okay? So the dominant alleles code for melanin. Okay, the recessive alleles code for no melanin. Okay, so for example, we could have individuals of intermediate skin color, right? And we could see what possible outcomes you could have in their offspring. So if they were all dominant, so if it was big A, big A, big B, big B, big C, they would be really dark, right? If it was all recessive, then they would be really light colored, right? So if we look at this, we can actually see if by chance, you do a Punnett square, but we're not going to do a Punnett square. I just want to show you that if by chance, like this one gets passed, right? This one gets passed, this one gets passed, let's see, little b, little b, little c, little c, little c, little c, right? We could have a gamete that is, excuse me, little a, little a, little b, c, c. Oops, sorry, the gamete, sorry. Oh, sorry. Okay, a gamete, A, B, C, A, B, C, right? So there's a probability that you would get all the recessive alleles coming together into the gametes, right? And so you could actually have an offspring, if this was the egg and this was the sperm, that would be this. So they would be very light in color compared to their parents. You could also see that there would be a probability of having like intermediate, right, skin color, and then you could produce an offspring that was very dark compared to their parents. So it is not true that if you have two dark-skinned individuals, that you could not possibly have a child that is light-skinned. The skin color is polygenic, and because of the way that the alleles separate, you could potentially have an offspring that is either lighter or darker than the two parents, okay? And that's a really important thing to understand, specifically when we start talking about races and ethnicity, because these two individuals are fraternal twins, right? So they're not identical twins, so they are as related to one another as they are sisters, right? These are their two parents, right? And this is them, and notice how different, obviously, the skin color is, right? So that can happen, okay? And in Europe, there was a big issue a few years ago because of the opposite. Actually, no, this is the same thing happening, right? Where we have darker skinned parents and light children. And these are people of the Roma ancestry, which are kind of derogatorily called gypsies in Europe, right? And so the Europeans were concerned. They thought that the gypsies and the Romas were actually stealing white children, right? They're stealing them. And so they were actually going into gypsy populations and removing white-colored children because they didn't understand the genetics behind skin color, right? So these guys they're actually the aunt and uncle, but they are this child's relatives, right? So it's really kind of traumatic. And the same thing happened in Africa during apartheid, where you could potentially have two white-colored people having a child that was dark-skinned, right? And so you could potentially have this happening. And this is actually a still from a movie that's called Skin. And it's a true story about two white people in Africa that have a daughter that is dark-skinned. And the government would not accept on her birth certificate that she was white, right? And so because she was labeled non-white, she could not have access to the same places that her parents could go, right? So she couldn't go to a white school. She couldn't go to places where only white people were allowed. And so what this suggests is that these individuals have a heritage, right, where they are able to pass on the dark skin gene to their offspring, right? And so skin color is a really important trait to understand, and to also understand that it is actually an adaptation that allows people to be protected against ultraviolet radiation. So why did we, if that is the case, what is the benefit of having light colored skin? Does anybody know? If dark skin protects against UV radiation, why would we have light-skinned individuals? Does anybody know why we evolved light skin? It has to do with a vitamin. What vitamin do we synthesize when we are exposed to sunlight? D, right? So people that are dark-skinned, right, and if they live in northern climates where there's not a lot of sun, they will actually become vitamin D deficient. So they would have to take vitamin D, and we actually now put it in milk, right, so that we can absorb it. If we don't get enough vitamin D, our bones do not grow properly, and we get a disease that's called rickets. Okay? So if we have light skin in northern latitudes, then we can synthesize vitamin D at lower light levels. So we can produce more vitamin D. So it's believed that the only reason why we have light-skinned populations, we all arose from Africa, is because we moved up to the northern climates and then our skin got light so that we could synthesize vitamin D. Okay. Okay, I did want to show you this. So this is a diagram that shows the variation, right? And so instead of being white and black skin color, like when we have attached earlobes and unattached earlobes, we now have continuous variation. And so we see that there is more variation in polygenic traits, and they also tend to have a normal distribution. So most people are intermediate. It's rarer to have really white skin, and it's rarer to have really dark skin. So most people are somewhere in the middle. So this is what is called a normal distribution. And so height is another example of a normal distribution. In populations, we tend to be of an average height and have very few people that are short and very few people that are tall. Okay? So polygenic traits also tend to be normally distributed. So we could write that we have continuous variation under polygenic inheritance, right? And we also have a normal distribution. Normal distribution, which looks something like this. This is the bell curve, right? So this would be skin color, and this would be number of individuals, and so as we increase skin color, it darken or lighten, right? Then somewhere, this is the average skin color, and most of the individuals are of an average color, or medium color. Okay. Any questions about that idea of polygenic inheritance? Continuous variation, normal distribution. Okay. So of the pedigrees that we have done thus far, none of them had to do with traits that were linked to sex chromosomes. And so when we look at the sex chromosomes, we have an X chromosome, which is big, right? And then the Y chromosome is much smaller, right? And this is actually what determines our sex, right? Our, whether or not we are male or female, in most cases, not all, right? And so the X chromosome has a lot more genes on it than the Y chromosome. So we would say that there is probably more X-linked traits than Y-linked. So X-linked traits are due to genes on the X chromosome. Right? So we should know XX is a female, right? And the XY is the male. Okay. So an example of this is red-green color blindness. So this is a gene on the Y chromosome. And it is recessive. So I would say it's X-linked recessive. So X-linked instead of autosomal. Autosomal means it's on a non-sex chromosome. Okay. So in this particular instance, we have to pay attention to who is male and who is female. So my X chromosome can have a big C on it, and that is normal. And my X chromosome could have a little c on it, and that is the color blindness. So that's the colorblind allele, okay? We do need to have to worry about Y just because of the difference between males and females. Because if you look at this, the genotype of a female could be this, could be this, or could be this. So yes, females can be colorblind. It's just more or less likely that a female, it's less likely that a female will be colorblind because she can actually be a carrier for the recessive allele and actually not have the traits. Males, on the other hand, could be X big C Y or X little c y. So notice how males cannot be a carrier. If they have only one copy of the dominant allele, they will have the traits, right? So that's why it's actually more frequent to see color blindness in males than color blindness in females, because they cannot be carriers, okay? So what we're gonna do is we're gonna draw a little pedigree here that shows the pattern of inheritance in a family. Okay, and I'm actually going to show how it could be passed down to a female. Okay, so I'm going to show it actually passing equally to males and females. Okay, so if I know that it's X-linked, I know that I need to put a Y wherever there is a box because males are squares, right? And so what I would do is I would put a Y here and a Y here. So that's the first thing I do. When I see a problem and it says sex-linked or X-linked, I always put the Y's in because that's really important place to start, okay? Now, this female, I know what she must be, right? She must be this combination. So I can write that in because she is colorblind. This male must be this combination. Right? This also male must be this combination. So what can I deduce about the mother? What must she be? A carrier. Excellent. Okay. So this is kind of a rare instance where we have a female who is a carrier for the gene, marries a colorblind male, right? And maybe they then have children, or they then have children that are colorblind, male and female. So that's the idea of X-linked or sex-linked inheritance is that you have to now pay attention, right, to which individuals are male and female. Now, could these individuals have a child that is a female, a daughter that is not colorblind? So I'm going to put right here. So a daughter that is not colorblind. Yes. And so remember, you just do the Punnett square exactly the same way. So this is my male, right? And this is my female, right? And I'll just put a circle there. That's my cross, okay? My times, okay. So then the males will produce, oops. The male would produce this possible combination. The female would produce this possible combination in the gametes, right? And so I just do my Punnett square. It's a 2x4, and it looks like this. Okay. So what's the probability that they would have a child that does not have, that is female, that is not colorblind? Okay, so is female. So if I say is female, you just ignore these males down here. She is female, right? So you say your baby's born, and you're like, oh, it's a girl, right? And you're like, oh, what's the probability? It'd be 50%, okay, because she's female. 0%, right? Oops, did I do that right? Oh no, 50% for male too. Right? Is that right? Yes, 50% for male too. To be colorblind. Okay, so that is sex-linked inheritance. Okay, so when we look at the X chromosomes are kind of interesting because females have 2 and males only have 1. So really, females have twice the genetic material as the male, and that is kind of a problem. So the problem is solved by what is called X inactivation. So if we start out as a single cell, or maybe this is an early embryonic cell, and I'm going to draw X1 and X2. Okay. So this is an early embryonic cell. So when this cell divides, something interesting happens early on, is that one of the X's becomes highly condensed and turned off. So it's random. So we would say maybe X1 is here, and then we have this little structure there, right? And then X2 is here, and then we have this little structure here. And then all the subsequent cell lineages from this embryonic cell is going to have X2 expressed, and this X1 shut down. Does anybody know what this structure is called? Has a name. This structure is called a bar body. So that is a bar body. So it's just the genetic material becomes wrapped tightly around the histone proteins, and it cannot be expressed. And so this used to be the way that they would test for sex. So like if you took a sample of my cheek cell and you stained it with just the right stain, you'd be able to see that I have one bar body in my cell. Okay? They don't do that anymore because they have better testing, and then we don't even really know what we're testing when we test for sex, whether it is presence of the Y chromosome or if it has to do with testosterone levels. Okay, so it's a little bit more complicated, but that's the bar body, okay? So this actually is the reason why calico cats are always female. And the calico that I'm talking about is the black and the orange, right? So if we look at in calico cats, we could have a female that has this, right? Where X big B codes for black and X little b codes for orange. Okay. And so because it's random, you get these weird patterns, right? Sometimes you see these really weird ones where there's like a line down their face and half of it's orange and half of it's black. But more often you see something that looks like this, right? And so what has happened is that the X early on during the embryonic development, these ones had the X big B turned off. So that was the chromosome that was condensed. And so they're orange here. These cells have the other X inactivated so that they are black. So we see this pattern on the surface of the skin, right? So that is a female calico cat. Sometimes you also see this, right? This is also calico, but there's another gene interacting. So there's another gene that causes the white patches, but the combination of black and orange on the body is due to X inactivation. Now there's a similar disorder in humans, but it's not color, so that pigment coloration, we don't really understand that yet. But we see examples of sweat glands and their distribution. So this is called anhidrosis, ectodermal dysplasia. Okay, big word for a distribution, an unequal distribution of sweat glands. Okay, so people that have this could have other symptoms. It seems to be a pleiotropic illness. But one of the symptoms is that due to X inactivation, some of their skin has sweat glands and some does not, right? And so you could theoretically have somebody, like when they sweat, they could have a line down their t-shirt where they have sweat glands on one side and no sweat glands on the other side, right? And so that is also an example of X inactivation in humans. Okay. Okay, so we are going to move on to the next chapter in your textbook, which talks about molecular genetics. And so when we talk about molecular genetics, we sometimes refer to the genome. And so the genome is all the genetic material in a cell. So most of that genetic material, almost all of it, is in the nucleus. There's a little bit in the mitochondria, right? We talked about that. Most of it is in the nucleus, and it's in the form of chromosomes. And we see that the genetic material is DNA. So DNA stands for deoxyribonucleic acid, DNA. Now, DNA is composed of nucleotides. So it's made of nucleotides. And the nucleotides that are in the DNA have letters associated with them. So A, when you hear the A, that actually means adenine. T is thymine. C is cytosine. And G stands for guanine. So the nucleotides, these are what are referred to, the A, T, C's, and G's. These are what are differ, and these are what are called the nitrogenous bases. And you can always identify them because they have a nitrogen atom in their structure. So these are the nitrogenous bases. So there's another part to the nucleotide, which is what is called the sugar phosphate. So I'll just put this as plus, the sugar phosphate. And they often call this the backbone. to the molecule. Okay. So nucleotides have a nitrogenous base, then they have a sugar and phosphate group. And that makes up the backbone of the DNA molecule. So if we look at a diagram of this from your book, okay, these are the nitrogenous bases. So you do not need to know the difference between pyrimidines and purines, but you'll notice that they differ in size. So the purines are actually larger, they're 2 rings, and the pyrimidines are a single ring. And then notice there's nitrogen. Okay, so if I pointed to this part of the DNA molecule, you would say that, you would say that this is the nitrogenous base. This is the sugar right there. And then these are the phosphates. That's the phosphate group. Okay, so we have a nucleotide being made up of that. Okay. Okay, so DNA also is double-stranded. And it has complementary base pairing. So I'll put with complementary base pairing. So the way that the bases pair is what is called Chargaff's rule. And this is obviously named after a person that figured it out, right, when they were looking at the structure. And Chargaff's rule says that A always pairs with T in the DNA molecule, and G always pairs with C. So if I gave you a sequence of nucleotides like this, A, C, T, G, G, C, right? This is one strand, right? You should be able to use Chargaff's rule to figure out what the second strand is going to be. And that's actually going to be part of your homework, okay? So the second strand, A pairs with T, C pairs with G, T pairs with A, C, C, G, okay? This is the other strand. Okay. Now, between the nucleotides on one strand, there are strong covalent bonds between these. But between the complementary base pairs, we have hydrogen bonds. So hydrogen bonds are designated with a dotted line. So you can draw like a dotted line between each of these. So that's a weak bond that can be broken. And so the DNA strand can actually break apart, right? And it does that when the DNA becomes copied, when we copy the genetic material prior to cell division. Okay, so if we look at the DNA molecule from your book, the diagram of it, oops, okay. So this shows the two strands, and then this shows one guanine pairing with cytosine, right? And then notice the sugar phosphate. That's actually the sides of the strand, right? And so this strand, the nitrogenous bases are sometimes called the rungs of the ladder. And then the sugar phosphate makes up the actual outside of the ladder, right? And the DNA molecule twists, and that is called the double helix. So the twisting of the DNA strands is called the double helix. And that was first written about scientifically by James Watson and Francis Crick. In 1953, they published their paper on the structure of the DNA molecule. So it's only been, not so long ago, 1953, that we first even discovered what the DNA was. Okay, so when we look at the DNA, it is the sequence of nitrogenous bases that codes for the differences that we see in proteins. So the sequence of nitrogenous bases along what we're going to call the template strand codes for protein. Okay. So when they talk about mutations, they say, well, one base changed. So instead of ATG, we have ACG. That means that that sequence changed so that it would possibly cause a change in the protein and could possibly cause a change in our phenotype. Okay? So it's the sequence of the nitrogenous bases, not the pairing, but the actual sequence along one strand that determines the differences that we see between individuals and between proteins. Okay? So when we look at the human genome, this is actually 3 billion base pairs long, right? So what does that mean? That means that there's actually 6 billion bases, because 3 billion pairs make 6 billion total, right? So that's a huge amount of DNA, right? So they actually, in the early 1990s and through mid-1990s through early 2000s, they were set about with the Human Genome Project, right? And what that was trying to do, the Human Genome Project was trying to do, is to take the human genome and figure out all the sequence of the A, C, T's, and G's, right? And they picked just an average person. Obviously, it's going to be different depending upon the individual, but they picked an average person to do the sequencing of the human genome, right? And what they found was kind of interesting. The Human Genome Project found that there's only about 20,000 genes in the whole human genome. And this is way lower than expected. And so we're going to talk about why that is, not today, but I just want to put that in there, that this is 20,000 genes. We predicted it was going to be like 200,000 genes, right? Instead of 20,000 genes. So we're going to talk about why that was the case. Okay. So a gene is just a piece of DNA that codes for a protein. Okay. So remember that the DNA is packaged, right? And so one of the things that we see when we look at a chromosome and we stain it, we can see that there are dark bands, and then there's that centromere that's really dark, and light bands, right? Usually the tips are darkly staining, right? So we have a pattern of stains, and this is what is called G bands, and they talked about that in your homework that you did on the karyotype analysis, right? And so this actually has to do with the difference in the chromatin and how it is packaged. So this darkly staining area is called, those areas are called heterochromatin. And they are tightly, the DNA is tightly packaged. And in this area, genes would be turned off. So genes are not coding for protein. Right. So it might just be, it's actually only 2% of our DNA is actually genes. 98% of it, we don't know what it does really. So it could be a part of our DNA that is that's not containing genes, and it's called heterochromatin. Okay. The lightly staining regions are not packaged tightly. and it is called euchromatin. So U means good. So euchromatin means good genetic material. Okay? And this is where the DNA is loosely packaged. So it's loosely wrapped around the histone proteins. Right? And genes are coding for protein. So in your book, they kind of show, oops, wrong way. They show this packaging, right? So this is the DNA molecule. This is the protein. So the DNA molecule, this in blue, wraps around these histone proteins, and then it gets tightly coiled, and then that is what makes up the chromosome. Okay? So we can kind of see at the chromosomal level where in the genetic material genes are actually producing stuff that like proteins. Okay. Okay. Oops, DNA replication. Okay, so DNA replication. This takes place in S. So that's the S phase of the cell cycle. So you should remember S and G1 and G2 and mitosis, right? So this is where this chromosome goes to this, right? So this is an unreplicated chromosome. This is a replicated chromosome. Now we have actually made a copy, an extra copy of the genes. And this has to occur prior to mitosis and meiosis. It has to take place prior to mitosis and meiosis. Okay? So the way that this works is we have enzymes that help in this. And so we have an enzyme called helicase. And helicase unwinds the helix. Okay. Then we have primase. And primase is what starts the replication. Okay. And then we have an enzyme called DNA polymerase. And so ACE, remember all these ACEs, that means it's an enzyme. Polymer means that it is an enzyme that is going to add nucleotides that are just kind of floating around in the nucleus of the cell to the single-stranded DNA after it has been unwound. So this adds nucleotides to the strand that is being copied, to the strand that is being produced. Okay. Okay, so in your book, they have a diagram of this. So we're going to kind of ignore that it's going differently in one direction from the other. You don't need to know that, but I'll kind of explain it. So here you can see that the DNA molecule separates, okay, and unwinds. And then what happens is that those hydrogen bonds between the bases separate. And so here you can see this is the DNA polymerase, right, the yellow box. There's also one of it up on top. And this is the primer that is added to the enzyme, or excuse me, to the DNA molecule. And what happens is it just gets built. So if I have a strand like this, right, what happens is I get a new strand built. So this is my new strand. And so I go from having just one strand of DNA to 2 strands of DNA, where this serves as a template. The polymerase just uses Chargaff's rule. So if there's an A here, it adds a T. G adds a C, right? And it just is moving down the length, okay? And then notice how it actually, when it goes the opposite direction, so the strand that is underneath, it actually has to do it in fragments. And then there's another enzyme called ligase, which puts those fragments together, okay? But the idea here is that one of the old strands serves as a template for the building of the new strand, okay? And it's all driven by enzymes. So those enzymes copy all of that genetic material, whether it's genes or not, right? We have to copy all 3 billion base pairs, right? And so that's a lot of copying. So sometimes mistakes are made, right? Oops, sometimes mistakes are made. You know, I'm gonna skip over the mistakes because we're gonna come back to that when we get to that in next week. Okay, so I'm gonna skip over this. Okay, so that was replication. So you can think about replication. If I draw a little diagram here, my DNA molecule is stuck inside of the nucleus. And so it makes a copy of itself, and this is called replication. So the DNA molecule makes a copy of itself. Okay. So this is in the nucleus. So I'm going to leave a little room here. I'm going to write more stuff here. So all that DNA replication takes place in the nucleus. Now, the DNA is stuck inside of the nucleus, so it cannot direct what is going on in the organelles. And so you have to make a copy of the information in the DNA, and that is what is called transcription. So the DNA molecule gets turned into a messenger RNA. And this is what is called transcription. So you could just, if you had that written up there, you could. That is called transcription. Transcription also takes place in the nucleus. Okay? So the messenger RNA is copied, is made in the nucleus. Now the messenger RNA is single-stranded, and it can be abbreviated as mRNA. Okay? So let's look at the mRNA. Let's take a look at RNA in general. So it's different, but it's similar. So RNA is single-stranded. Okay. It also, instead of T, we have U. So uracil, not thymine. So uracil is the equivalent of a base, a nitrogenous base. RNA does not have T's, it has U's. For some reason, I'm not sure why. Okay, but it actually makes it easier for us to identify what's messenger RNA, what's RNA, and what's DNA. Because if there's T's in the sequence, you know that has to be DNA. If you see a U in the sequence, you know that has to be messenger RNA. Okay. So transcription is where we have the template strand. So I'm going to say this is my template strand, DNA strand, and it has sequences of bases. So it could have A, T, G, T, C. Okay. So we have to take that information in the DNA and we have to transcribe it. We have to make a copy of it. It's in the same language. So it's same nucleotide language. So we use Chargaff's rule, but the messenger RNA that's going to be produced is going to be A is going to be U. So notice A would normally pair with T in DNA, but it pairs with U in the messenger RNA. T pairs with A. G pairs with C, T pairs with A, C pairs with G. Okay, so this is messenger RNA. Okay, Now, one of the reasons why it's called messenger RNA is it leaves the nucleus and it travels and carries that information out into the cytoplasm and it hooks up with a ribosome. Okay, so the messenger RNA leaves the nucleus and attaches to a ribosome in the cytoplasm. And the next stage is taking that information that is in that sequence of A, U, G, for example, and turning it into a sequence of amino acids in a protein, okay? So that is the process of translation. So I'm gonna skip, we'll come back to this in a little bit, but to translation, okay. So translation is where we go from messenger RNA to protein. And the reason why it's called translation is we go from A, G, U, C, right? That's nucleotides, those are nitrogenous bases, to amino acids, okay? So those are two different languages. We have the nucleotide language and we have the amino acid language. So remember that that is translation. That makes sense because it's not just making a copy, it's not just transcribing it, it's actually changing the language, okay? That's why it's translation. Okay, so this takes place at the, in the cytoplasm, right? This is in the cytoplasm. And it involves another RNA molecule, which is called a transfer RNA. This is abbreviated as tRNA, okay? The transfer RNA is a small molecule comparatively, it's very small compared to messenger RNA, right? And it has a 3-letter sequence, which is called the anticodon. So this is a 3-letter sequence of nucleotides that can bind to the messenger RNA. Right, through base pairing. That's the anticodon. We'll talk about what the codon is in a second. And then it has a binding site for a specific amino acid. Okay. It's a binding site for a specific amino acid. So oftentimes it's drawn looking something like this. So we have our tRNA and we have 3 letters. So let's say we have GUU. Okay. And then we have an amino acid. So that's my amino acid. This is my anticodon. Right? My messenger RNA is a big long strand. Right? And so what would be the complementary sequence of messenger RNA to the GUU? G pairs with what? C. U pairs with A. Right? Okay. So what this tells me is that in this particular position on the messenger RNA, we need to add a specific amino acid that is CAA. And there's a table in your book that kind of goes along with the homework I'm going to give you. And CAA corresponds, I always look it up because I don't have these memorized, to proline. So this is proline. And it's abbreviated as PRO, proline. So this right here is my codon. Okay. So the messenger RNA has codons, the tRNA has anticodons and amino acid. So what happens is that this all takes place with enzymes, and like surrounding this is a big ribosome. So this would be my ribosome. And what happens is that ribosome reads the messenger RNA, and the appropriate tRNA comes in, and then that amino acid that should be in that position gets transferred to the growing chain of amino acids that is being produced. And so essentially, this is protein synthesis, right? So we're protein synthesis. So the information from the DNA gets transcribed into messenger RNA and then translated into a protein of a specific sequence so that when it folds, it has a specific function, okay? So in your book, they have a diagram that's similar to the one I just drew, right? So the blue thing is your ribosome. This is my tRNA, and then this is showing the growing chain of amino acids, right? And then this is the messenger RNA. Okay. Now, how do I know which codon goes with which amino acid? So the answer is this table right here. I do not memorize this table. So this will always be given to you. And in your homework, I put it on the back of your homework. But it's also in your textbook, okay? And it's also online. So this is the genetic code. So this tells me messenger RNA codon equals what amino acid? Right, so you can't use DNA. Notice that there are U's, right? So first you have to transcribe, then you have to translate. Now you'll notice something interesting about this is that some of the codons say stop. So there is punctuation in the genetic code. So if you get to a stop codon, then translation just stops and the protein is cut off and then the protein is finished. So genes generally have a start and they have a stop. So they have a sequence that tells them to start and the sequence that tells them to stop. It's kind of like a capital letter and a period, right, in a sentence. So it tells it how to read it, okay? The other really important thing to realize is that this is a universal code. So what that means is that all organisms on this planet use it, right? So that's why you can take a gene from a jellyfish and put it into a rat, right? And the rat will glow like a jellyfish because it is reading the gene because the genetic code is universal. So it doesn't matter what organism cell you put it in, an organism cell will be able to read the genetic material. It's also why viruses can insert their DNA, and our cells make more viruses, right? They make viral proteins, right? Okay, so homework. So this will be due on Tuesday. All you need to do is apply Chargaff's rule where it's appropriate, remembering where you use T's and where you use U's, and also using the code. No, that's I'm sure. We just have class on Tuesday. We're going to be doing DNA fingerprinting on Tuesday. Okay, so before you put it away, I just want you to look at it for a second. Okay, so to put strands, you would then, I know there's some, you can pass them up if there's extras. You guys give. Did you guys get enough down there? So let's just go through, I'll just do the first one, okay? So DNA templates, that would be CAA, right? CAA then gets changed to GUU. Are you following along with this?
Wait, how does C go to U?
Okay. CAA is the DNA template. Okay. From the DNA template, I go to GUU. Because A would normally pair with T, but it pairs with U. Okay. Then I would go to this table, right? What is GUU? VAL. So I write VAL. Okay. And then what is, if I look at the messenger RNA codon, what would be the anticodon? So GUU, apply Chargaff's rule, CAA. I totally lost everybody. Okay. So the tRNA anticodon is just taking messenger RNA codon and applying Chargaff's rule. What would it pair with? Yes. Okay. So just everybody write this first one on their page. I'm giving you the answers here. Okay. So it is GTT, right? Template CAA. Messenger RNA codon GUU. Amino acid VAL. And then I go CAA because that would be my tRNA. So my tRNA, right, would have this as its anticodon, and then it would have VAL on it, right? So ignore that, okay? Okay. And did you get it? Sort of? Okay. So this is reading the genetic code. Sickle cell anemia. Okay. Yeah. No. Just no problem. Yes. Yeah.